Hi everyone, thank you all so much for joining this session. Today we're really excited to be welcoming Caitlin Davis. Caitlin Davis joined Yale in January 2020 as an assistant professor of chemistry. Her field of research is biophysical chemistry with a focus on quantifying biomolecular interactions inside living cells. Davis's experimental approach makes connections between molecular mechanisms and cellular function through time-resolved spectral imaging at multiple scales, from in vitro to single-cell to whole organism. Her research has been published in journals such as Nature Communications, Journal of the American Chemical Society, Chemical Science, and Biochemistry, among others. Before coming to Yale, Davis was a Center for the Physics of Living Cells postdoctoral fellow at the University of Illinois. She received a PhD in chemistry from Emory University and a BS in chemistry and mathematics at the University of Michigan. Her talk today is entitled Protein Folding from the Test Tube to a Living Organism. So without further ado, I will hand over to her now.
So I wanted to first thank the organizers for inviting me here today to share my research on protein folding from the test tube to a living organism. So first I wanted to get started with talking a little bit about proteins. What are proteins? So I know that we have a very diverse audience and I just wanted to get everybody on the same page. So proteins are very important molecules in our cells. They're involved in virtually all cell functions, and every protein inside the body has a different role. Antibodies are proteins that protect the body from germs. Hemoglobin is an example of a protein that carries oxygen from the lungs to the rest of the body. And all proteins are made up of amino acids. Different proteins differ in chain length and which amino acids comprise them. These chains fold into structures that are made up of coils called alpha helices, shown here, or into sheets called beta sheets, shown here. And these alpha helices and beta sheets pack into a specific three-dimensional structure. And in order for the protein to function, it needs to be correctly folded into this three-dimensional structure. If the protein misfolds, it will lose its ability to function, and this has been implicated in misfolding diseases like Alzheimer's, Parkinson's, Huntington's, and even mad cow disease. So how do proteins fold? Anfinson's dogma states that the three-dimensional structure of a protein should be determined solely based on its amino acid sequence. So we should be able to predict the three-dimensional structure of a protein based solely on that sequence of amino acids. But polypeptide chains can adopt many different three-dimensional structures. Every amino acid can access 3 different conformations. So if we had a 100 amino acid protein with 3 conformations available per amino acid, that would be 3 to the 100th or 10 to the 48th distinct conformations. And if the protein were able to test 10 to the 13 of those per second, it would take 10 to the 27 years, or longer than the age of the universe, for the protein to fold. But proteins are able to find this three-dimensional structure much, much faster, in under a second, often as fast as microseconds. So proteins are thought to fold on a free energy landscape shaped like a funnel, with the lowest energy state being the biologically functional native state. And the energy landscape tells us about protein folding. Smooth funnels lead to fast folding, whereas rough funnels with bumps lead to slower folding. Some proteins may get trapped in misfolded states, or local energy minima on this rough energy landscape. Misfolded states have been implicated in protein misfolding diseases like Alzheimer's, Huntington's, and Parkinson's disease. So in order to understand how proteins fold, we need to be able to map out this energy landscape. And mapping the energy landscape has required close integration of computation and experimentation. So experiments can test computational predictions, and computers can guide and interpret experimental results. Yet until recently, a divide existed between what could be computationally predicted and what could be experimentally tested. So computers can only fully fold the smallest proteins that fold in under a millisecond. but experiments could only test proteins that fold in over, say, 10 microseconds. And in my lab, we use laser-induced temperature jumps to bridge that timescale. Using a laser-induced temperature jump, we can monitor protein folding on the nanosecond to millisecond timescale. And this has really given us a lot of information about the early events in protein folding, so we can understand how misfolding is initiated. So this conference is called A Timeline of Science, and I thought I would give you a timeline of this temperature jump approach. So Manfred Eigen won the Nobel Prize in 1967 for studies of extremely fast chemical reactions by disturbing the equilibrium by means of a very short pulse of energy. So this is essentially a temperature jump. He said that you could use a laser to quickly change the temperature of a solution, and that would enable you to study very fast processes. And we can see that in what's called the Van Hoff equation. So this shows you that if you perturb the chemical equilibrium by temperature, it only requires that the reaction have a dependence on enthalpy in order to see a change. And the advantage of this is that it only requires the transfer of energy from that heating source to the solution, so you can do it really fast. It can be very difficult to find a T0 in your sample or the initial time, and in this case the sample starts at equilibrium, so you're able to initiate reactions using this, and your results are directly related to enthalpy. So in this equation, this delta H is enthalpy, and so our observations are related to that. Now, in biophysics or protein folding, this really started in the mid-90s with this idea that we would be able to merge computation and experiment. And these are examples of some of the small peptides that we wanted to fold. In 1995, we were able to fully fold these small peptides, but it wasn't until 2011 that the computer simulations were able to catch up and really fully fold all of these peptides. At the same time, we realized that the next frontier in this field is to study proteins in their native cellular environment. It's not enough to just study them in a test tube. We want to push things in cells. So in 2010, the first experiments were conducted inside cells in the lab that I did my postdoctoral research. And while I was there in 2019, I developed the first method for doing these types of studies in a whole living organism. And so I'm going to tell you a little bit about that today. So in order to study proteins in cells, we needed to have an approach. And our approach is to do in vitro and in-cell experiments hand in hand. So what I mean by that is that we first characterize a specific protein of interest in a test tube without making any modifications to it. And there are intrinsic things about the protein that we can monitor. For example, this is an amino acid called tryptophan that will fluoresce. But there are lots of proteins inside cells that have tryptophan. So we're not going to be able to use that to look at this protein inside a cell. So what we do is we add fluorescent labels to it that are different colors so that we can see it inside our cells. So for example, we could use a fluorescent protein label, this is GFP, or we could use an organic dye and attach that to our protein. So here, this is an organic dye label. And what we do is we attach these dyes and check and make sure that they don't change the behavior of the protein in the test tube. We have a strategy for observing the protein inside our cells that's not disrupting its behavior. We then repeat our experiments inside the cell. This cycle is closed when we adjust our test tube conditions to better mimic what we observe in cells. And so the story that I'm going to tell you about today has to do with phosphoglycerate kinase, or PGK. And PGK is a great model for our protein studies because it's ubiquitously expressed across many different cell types. So that means that PGK is found in many different types of cells, and this is because it's involved in step 7 of glycolysis, which is an important process. Sugar is produced. It also folds independently of a chaperone. So some proteins require other proteins to assist with their folding, and PGK doesn't require that to fold, so we can study it in a test tube without any other molecules. we did make some mutations to the protein, and this was to destabilize it to a temperature we could study it in cells. So cells can only really survive up to around 55 degrees. So we wanted to destabilize it down to around 37 degrees where we could study its folding. And our labeling strategy for this was to label one side with the green label called GFP and the other side with a label called M cherry. And these dyes, when they're close together, if you excite the green one, it's actually able to transfer energy to the red one in a process called FRET, or fluorescence resonance energy transfer. So we use the end-to-end distance to tell us about folding. When the protein is folded and the dyes are close together, we'll see red. And when the protein is unfolded and the dyes are far apart, it will look green and it will be unfolded. And I will show you a video of that in a couple slides. So before we do any experiments in cells, we need to characterize our protein. And we do that in a bunch of different ways. The first one is by circular dichroism. So this tells us about the secondary structure, or that alpha helices or beta sheets that are in our structure. And this is the way that the secondary structure spectrum looks at different temperatures, from low temperature to high temperature, from blue to red. And this is consistent with beta sheet structure, which is these large labels that we added, the beta barrels. So that's what that is picking up. But if we follow the minimum wavelength here, we see that as we increase the temperature, we see a loss in structure. So what's happening is our protein is unfolding between 30 and 40 degrees, around 35 degrees. We still have this structure at our highest temperature because our labels are still folded, so it's picking up that structure. We can also use tryptophan fluorescence, so that intrinsic fluorophore that I told you about, to monitor folding. And what happens in this case is that when the protein is folded, that signal is quenched, and then when it unfolds, we'll see an increase. And again, we do see an increase in fluorescence between 30 and 40 degrees. So that tells us that the protein is unfolding around 35 degrees. And then finally, we can use that FRET. So here I excite at the green wavelength, which is right here, and we can see that there's a transfer of energy in the blue one over here. So the small peak is the red. And as we raise the temperature and the protein unfolds, it's unable to transfer energy from the green to the red, and the signal goes away, and we see an increase in the green signal. And we monitor this by plotting the donor over acceptor ratio, or the green over the red signal. So when the protein is folded, we have more red in this ratio, so we have a lower D over A value. And when it unfolds, we have more green. So we again see this transition between 30 and 40 degrees. And if we fit this to an equation, we can get a melting temperature of around 35 degrees, which is where this midpoint of the reaction is. So this is all great, but we want to be able to do these experiments inside cells, and we can't put our sample into these instruments. So what we do is we actually do our experiments on a microscope. So here we have our microscope, and we can express our protein inside a cell on a cover slip in a slide, or we could put our in vitro sample in here. And then we can we can excite our green and then monitor changes in green and red signal on a camera. And we can slowly raise the temperature of the entire sample stage by slowly heating it up. And so here I'm comparing results that we conducted in vitro and in cells. So there are two things that should be immediately apparent. The first one is that the protein is more compact inside cells compared to in vitro. So these D over A values are lower. That means that their protein is more compact. And the second one is that the midpoint is slightly shifted inside cells compared to in vitro. So they're a little bit more stable in cells. Finally, at high temperature, we do see some of these negative effects. So we see some aggregation at high temperature. But we don't just want to know about the thermodynamics of folding or the midpoint. We actually want to know about kinetics and dynamics. And so that's where we use our laser-induced temperature jump. So if we quickly heat the sample on a timescale faster than the protein is able to actually fold into this conformation, we should be able to monitor the folding process. So here I would do something like raise the temperature from 30 to 35 degrees. And then with my camera, I can monitor the change in the protein signal as it relaxes to this new equilibrium up here. So this is a video of that, and I've expressed my fret-labeled protein inside this mammalian cell. So the red here is because the protein is starting from a folded state, so it looks more red. And this is expressed in the cytoplasm of the cell, so that's why the nucleus is gray. We'll start a temperature jump at T0 here and turn it off at the high T. And as it relaxes to this new higher temperature, we will see the color of the cell change. So here I do my temperature jump and then monitor as it relaxes to this new equilibrium at the higher temperature. And once it's fully reached this equilibrium, we'll release the temperature jump and monitor as it relaxes back down to the initial temperature. And what we can see is that this is a completely reversible process. So the initial and final signal are the same. So the protein is reversible between those temperatures. It can fold and unfold. And for a two-state process, we expect to see single exponential kinetics. And indeed, we can map a single exponential kinetics onto this. So what do we see when we compare in vitro and in cells? Well, the folding dynamics are unchanged in cells. If we compare in vitro, our circles, to in cell, we see that this is almost identical. So our classical in vitro measurements in a test tube were able to adequately reproduce the folding dynamics and mechanism. But if I just look at the equilibrium measurements, those thermodynamics, looking at the midpoint, I saw that the midpoint was slightly higher inside cells and the conformation was more compact. So structure-dependent measurements should be, like catalytic activity, should be carried out in the presence of Crowders. And if we take this PGK and we do measurements in the test tube in the presence of Crowders, we can improve its activity by about 40-fold. So the best approach inside the test tube depends on the question that's being asked. And the next question that we asked was, well, if measurements are different in this cancer cell line that we used than in the test tube, then what happens in differentiated tissues? Because the local environment of every tissue in your body is different. So do observations in cancer cell lines translate to differentiated tissues? We developed this customized pipeline for quantifying protein thermodynamics and kinetics in living zebrafish larvae. And don't get too overwhelmed. I'll go through each part step by step. We chose zebrafish for these studies because they are transparent. So it's possible for us to see the fluorescence inside this organism. And secondly, because they're cold-blooded. So they're sensitive to changes in temperature. So we should be able to do the same types of temperature-dependent measurements. So what we do is we modify their DNA at the single cell stage. Then 2 days post-fertilization, we observe expression of our fluorescent labeled protein in single cells within the organism. And we use that same microscopy approach to monitor individual cells within the zebrafish and get out their stability and kinetics. So some of you might have heard of CRISPR-Cas9. That's a method where you can genetically modify the entire genome. But here what I'm doing is I'm using a modification method that is not specific to vertebrates. So there's no match for this enzyme that's going to help, or meganuclease that's going to help us insert the gene into the zebrafish. Because it doesn't have a specific binding site, incorporation is slow and it occurs beyond the single cell stage. So we only get a few of our zebrafish expressing our fluorescently labeled protein. And we inject this together with our transgene cassette, and this contains all of the information that the zebrafish needs to express the protein. So it has a promoter to tell the fish what tissue to express it, our FRET PGK gene, and then a polyadenylation signal. So this tells it to turn off or to stop expressing the protein. And we compared expression in 4 different tissue types: in the muscle, the skin, the eye lens, and the notochord. And we did this because these represent very different environments. So the muscle is interior, the skin is exterior and needs to respond to external stress, The eye lens is unique because it's the most crowded tissue type in an organism. So in order to build that lens, proteins are packed really tightly. And then the notochord runs along the spine, and it's dominated by an enlarged vacuole, or like a big water sac. So these are all pretty different environments. And 2 days post-fertilization, this is expressing our FRET PGK in a single cell in this living fish. And at 2 days post-fertilization, the fish are not yet mobile. So we position them on the slide cellulose and place them under anesthesia for imaging. We then create our little chamber by using a spacer and a slide on top. And we can see fluorescence of these individual cells. So we do 2 types of measurements. The first kind, we just slowly warm up the entire aquarium here. And the second time, we would do our temperature jump experiment just on these individual cells. So first, we wanted to make sure that we had promoter-dependent localization of the FRET PGK. And so we can use reference histology to confirm that. So we have expression of our protein in the muscle, the skin, the notochord, and the eye lens. So this is pretty cool. And we did 10 measurements for each tissue type. And what we see is that in general, the fish can maintain homeostasis. What does that mean? It means that in general, the stability of the protein was very similar in all of the fish. So in the muscle, skin, and the notochord, the stability of the protein was around 38 degrees. But in the eye lens, it was further stabilized with a stability of almost 41 degrees. So the protein is stabilized in the eye lens compared to other tissues. And what I did is I normalized this data and I compared it to measurements that I collected in the test tube and in our cancer cell line. And here it is. So this is what I showed you earlier. And then this is just normalized between 0 and 1, so we can easily compare everything. And I see that all of my tissues in the cancer cell line are more stable than the test tube. And also the slope of this is a little bit different. So that suggests that folding is more cooperative in cells, which means the mechanism might be slightly different, that some of those chaperone proteins may assist with folding. Now, we also looked at the kinetics in cells, and what I saw is that the kinetics are unchanged inside the zebrafish compared to the in vitro and in cells. But the eye lens, the folding was slightly slowed. So how can we explain differences between in vitro and our different tissues? Well, the cellular environment is very crowded and sticky. So only 60 to 70% of a cell's volume is made up of water. And this is what our normal test tube environment looks like. But another 8% is made up of electrolytes and small organic molecules and metabolites. The other 22% is made up of nucleic acids and proteins. So inside cells, there's a tug of war between steric crowding interactions and non-steric sticking interactions. And the steric crowding interactions are generally stabilizing, and these non-steric sticking interactions are generally destabilizing. So steric crowding, you can think about if you were walking through a crowded train station or a crowd. You wouldn't want to walk around with your arms out. You would have them close to your body. A protein is the same way. When you put a protein into a crowded environment, it's going to be more compact because those extended states are destabilized. And then the sticking interactions has to do with the surface. So you can think about the surface of the protein as being covered with patches. Some of them could be like Velcro, where they would stick to things, and then other parts would be like oil or really slick, and so they would slide away. And so those will change the stability, and they'll have different impacts on the folded and unfolded state because both have different surfaces. So that could stabilize or destabilize the folded and unfolded state differently, resulting in a different stability of our protein. So our hypothesis was that this PGK protein was stabilized in the cytoplasm because it evolved to function in the cytoplasm. And if we studied a protein that was not evolved to function in the cytoplasm, that it would be destabilized. So we compared the folding of PGK in the test tube and in the cell to another protein called variable major protein-like sequence expressed, which is a mouthful, VLSE. And VLSC is the outer membrane protein. It's expressed in the cytoplasm, and then it has to be destabilized in order to be transported and then refolded in the outer membrane where it folds and functions. And so when we look at the stability of that protein VLSC, it is indeed destabilized in cells. So you can see here that in vitro, it has a very similar stability to PGK in the test tube, but in cells, it's more expanded. So the states that we see are more expanded, and it's destabilized. So these are great systems for understanding those sticking and crowding effects. And I did a series of experiments under different conditions, mimicking crowding and sticking. So we can mimic this crowding interaction by introducing inert macromolecules into the solution, and it's called FICL. So it's a big long chain of polysucrose, and as we increase the amount of that, we see that we're compacting the protein. And if you look closely, you can also see that it's affecting the stability of the protein. And then we used really dilute cell lysate to introduce a bunch of the other things that could be affecting the sticking. So that can be the electrostatics, the amount of the ionic strength in the cell, or the salts. It could be some of those small metabolites, or it could be surface interactions with other large macromolecules. So we use dilute cell lysate, so we have some of the other macromolecules that would have been in solution in the cell, but just not at a concentration where it would be crowding the protein. And when we do experiments in different and titrate in an increasing amount of our sticking mimic, we see that both proteins expand as you increase the amount of that sticking. And here the closed circles are VLSC and the open circles are PGK. And that increase, or that expansion of the protein, is all due to the ionic strength, or the salts. So here this is mimicking the amount of salt in the cell around 200 millimolar potassium chloride. And we also see that in our lysis buffer. So the lysis buffer has some of those small metabolites and fatty acid change, just not the protein. It's also expanded. So just the ionic strength of the solution is causing this expansion. And that almost mimics the expansion that we saw for VLSE, but it can't explain the compaction of PGK. So if we do experiments in our crowding mimic, we see that here PGK is very compacted by our crowding mimic, but VLSC is relatively unaffected. And I'm sorry, I didn't explain here. Our axis here is the donor over acceptor ratio. So we're just looking at compaction here. And what you see is that this compaction almost extrapolates in cell PGK. So what this is telling us is that PGK is more sensitive to crowding and VLSC is more sensitive to sticking. So that could explain the difference inside the cell, that the crowding is stabilizing PGK and the sticking is destabilizing VLSC. And we can do the same thing to look at this thermal stability. So I just calculated what is the stability of the protein at different concentrations of FICL. And in both cases, the proteins are stabilized. And this makes sense because the extended conformations are destabilized in both cases by crowding, just purely crowding. And that can't be explained by salt. And we see that our lysis buffer, those small metabolites and fatty acids, have different effects on each protein. So PGK is slightly stabilized and VLSC is destabilized. And that actually mimics what we see in cells. So PGK is stabilized and VLSC is destabilized. But this effect is even more apparent when we look at our sticking mimic, cell lysate. So here you can see as you increase in the amount of cell lysate, that PGK is going to be stabilized and VLSC is going to be destabilized. So if you want to predict the stability trend in cells, you could use just your sticking mimic to predict whether the protein would be stabilized or destabilized. And we realized that we had something that looked like it could reproduce the cellular environment. If we just add our measurement in lysis buffer to our measurement in FICL at around 150 mg per mil, we will almost perfectly reproduce our in-cell experiments. And so what we've done since this is in a paper that was published in February of this year, is we've shown that a mixture of FICL and cell lysate is able to actually reproduce these measurements for both proteins. So this is something that if you wanted to reproduce the cytoplasm of a mammalian cell, that might be able to reproduce that that condition for you in a test tube. So we also looked at the kinetics, and I saw that as I increase the amount of cycle, that PGK is initially sped and then slowed. And here is where we use simulations to help explain this. So it turns out that crowding initially compacts the protein and assists with folding, but at high amounts of crowding, the viscosity of the solution or the thickness of it actually slows the folding, and that VLSE is unaffected here. And we see the opposite for cell lysate. So PGK is unaffected and VLSE is slowed. So that tells us why PGK is relatively sped in solution or has a similar folding rate, and why VLSC's folding is slowed in solution. So now if we go back to that slide that I showed you, and this is my last slide because I know time is short and you guys want to ask questions, that we had seen that in the eye lens was most impacted because it's the most crowded. So the eye lens is stabilized the most because of crowded environment, and its folding is slowed because it's most viscous here. So we can explain our tissue experiment using these in vitro measurements. And so I just want to quickly thank, this was work that I did in my postdoc, the Grubela lab, and in particular the people who contributed to this project. Ruth Pei Feng, who is shown here, worked with me on the zebrafish project, and Jonathan Deutsch worked with me on the recent measurements that I mentioned. My postdoc was funded by the Center for the Physics of Living Cells at the University of Illinois and the NSF. And I'm just started as an assistant professor at Yale in January of 2020. If you want to check out my current research, you can look at my website or follow me on Twitter. And I do have graduate positions available. The application deadline is December 1st. So if you're looking for graduate school, check out Yale. And I'd be happy to answer any questions. Okay.
Thank you so much, Professor. I hope you can all hear me now.
Yes.
Cool. And thank you so much for such a fascinating lecture. It was so interesting to hear about all of your research into protein folding and stability, its applications as well. So thank you so much. We've got a couple of questions. So the first question was, how do you ensure that adding the fluorescent labels does not affect protein folding?
Right, so that's a great question, and that's why at the beginning of our studies, we use this approach. So it actually turns out that on PGK, when we use the fret label, the green and the red fluorescent protein, that the red actually does slow the folding. So it's important to fully characterize the protein in the test tube first, and we can study its folding, look at the stability and the kinetics, and then do the same thing with our fluorescent proteins attached to it. And if you change out that red fluorophore to something smaller, so this would be like a reash label, which is red, it recovers its folding. So in that case, the slight slowdown of PGK that we observed was due to the fluorophore like getting stuck in the cell and slowing its folding. So it is something that we have to consider in the design process. Does that make sense?
Yeah, that does. Thank you. And the next question was, why did you choose zebrafish as your model?
Right. So we selected zebrafish as a model because it's the simplest vertebrate that is used as a model for studies, and we wanted something that was a vertebrate so it would be more similar to mammals when we're looking at the tissues. So zebrafish share a lot more of their genome with humans than, say, C. elegans do. And when we're looking at specific genes in the zebrafish and in that environment, there are a lot of different models that can model disease like human disease. So if we wanted to move forward and study breast cancer, there's a model for breast cancer in zebrafish. So that was another reason why we were interested in zebrafish. And then the other reasons are that they're transparent, so we can see the fluorescent protein within it, and that they're cold-blooded, so they'll be sensitive to those temperature changes. And since all of our measurements use temperature to study the protein, it's important that the organism be able to respond to a temperature change.
So you noticed that the protein was stabilized in the islands. Why do you think this was? And do you think a similar pattern would be seen in any other tissues with similar characteristics to the islands?
Yeah, so I didn't give you the actual numbers for the islands. I apologize. So um, the rate of what we call macromolecular crowding is measured in milligrams per milliliter. And in a normal like tissue, like your muscle, the amount of macromolecular crowding is usually around 150 to 200 milligrams per milliliter. The islands has the highest rate of macromolecular crowding. It's around 500 milligrams per milliliter. So it's quite a bit more crowded than the other tissues. And I would predict that if you looked at other similar tissues that were crowded, you would see that. Something I also didn't mention that I think is interesting about the islands that's a future direction is that our islands is made up of a protein called alpha crystalline. Um, and alpha crystalline is actually a chaperone. It's not a specific chaperone for PGK, but it's possible that it could be assisting with the folding. So that's a future direction for this project, is to see if maybe that alpha crystalline that's already in the islands is affecting the folding of PGK.
Thank you. And the next question was, could these results be applied to human cancers?
Yeah, so I guess it depends what you're interested in cancers. So something that I am interested in is the environment in cancer is more oxidative, or there's lower oxygen levels, and that can affect disulfide bond formation in proteins. So it's possible that a cancer cell line, the folding would be affected because the amount of oxygen available is different within that environment. And so if we take a protein that's very sensitive to formation of disulfide bonds, it could be that the folding is different in a healthy tissue than in a cancer environment. And that's actually a project that we're working on right now in our lab, is how to adjust the oxygen levels in the tissue and monitor folding under those conditions. And I think we have some, we might see something interesting there.
And in relation to that, you were talking about the stability of molecules sticking. Does the differential expression of adhesion molecules on cancer cell surfaces affect this and lead to a changing pattern, or is this sticking intracellularly?
So the sticking that I was looking at was intracellularly. Um, if you were studying a protein that was an outer protein, then maybe. But all of the measurements that I did were inside the cytoplasm.
Okay, thank you so much. I think that's all of the questions we have. So once again, thank you so much for such an interesting lecture. I really enjoyed it, and I'm sure everyone else has too. So yeah, thank you.
Great, thank you for having me.